Hello viewers, welcome to our channel. In this video, we will discuss some important questions on transcription. In my previous video, we have already discussed some important questions on transcription. The link is shared in the description box. For CSIR NET, ICMR NET, GATE, NEET exams, the questions regarding this topic is very important. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. During post-transcriptional modification of H and RNA, that is heterogeneous nuclear RNA, capping involves option 1 addition of polyadenylation at 3' end, option 2 addition of polyadenylation at 5' end, option 3 addition of 7-mg, that is 7-methyl guanosine cap at 5' end, and option 4 is addition of 7-methyl guanosine cap at 3' end. And the correct answer is option 3, that is addition of 7-methyl guanosine cap at 5' end. Now let's discuss about capping. Capping is the process of addition of 7-methyl guanosine cap triphosphate to the 5' end of H and RNA. It protects the RNA from nucleases. It is essential for mRNA stability, enhancing mRNA processing, mRNA transcript, and translation. After successful capping, an additional phosphorylation event initiates the recruitment machinery that is necessary for RNA splicing, a process by which the introns are removed to produce a mature mRNA. Next question. The UTRs are required for efficient translation, which are present on option 1 3' mRNA, option 2 5' mRNA, option 3 D loop of tRNA, and option 4 both 1 and 2. And the correct answer is option 4, that is both 1 and 2, that is UTRs are present in both 3' mRNA and 5' mRNA. In molecular genetics, an untranslated region refers to either of two sections, one on each side of the coding sequence of an mRNA strand. If it is found on the 5' side, it is called the 5' UTR or the leader sequence. If it is found in the 3' side, then it is called the 3' UTR or the Taylor sequence. The UTR plays a critical role in translation termination as well as post-transcriptional modification. The 5' UTR is a sequence that is recognized by the ribosome, which allows the ribosome to bind and initiate translation. Next question. What initiation and termination factor are involved in transcription of prokaryotes? Option 1 is sigma and rho. Option 2 sigma and beta. Option 3 sigma and gamma, and option 4 beta and rho. And the correct answer is option 1, that is sigma and rho. Now let's discuss about sigma factor. The sigma factor is a specificity, also known as specificity factor. It is a protein that is needed for initiation of transcription of bacteria. Although once that stage, that is the initial stage, is finished, the sigma factor is dissociated from the complex and the RNA polymerase continues the elongation on its own. The sigma factor is the transcription factor of bacteria that enables specific binding of RNA polymerase to the promoter region of the gene. So sigma factor is mainly responsible for the specific binding of the RNA polymerase to the promoter region of the gene. It is homologous to archeal transcription factor B and to the eukaryotic factor TFIIB. Next, the Rho. Rho is a protein that is capable of binding single-stranded DNA and it terminates the transcription. Which polymerase produce the transcript that are 5' capped, polyadenylated, spliced to ultimately be translated into the protein? Option 1 is RNA polymerase 1. Option 2 is RNA polymerase 2. Option 3 is RNA polymerase 3. And option 4 is all of the above. And the correct answer will be option 2, that is RNA polymerase 2. Here the question is, which polymerase produce transcript? That is ultimately spliced, first 5' capped, polyadenylation and spliced, ultimately to be translated into protein. So, but the transcript is mRNA, and this mRNA is produced by RNA polymerase 2. The RNA polymerase 2 produce the mRNA that are 5' capped, polyadenylated and spliced to ultimately be translated into a functional protein. Whereas the polymerase 1 in eukaryotes is to transcribe ribosomal RNA, with the exception of 5S RNA. That is, RNA polymerase 1 does not transcribe the 5S RNA. Instead, this 5S rRNA is transcribed by RNA polymerase 3. RNA polymerase 3 not only transcribe the 5S rRNA but also transcribe the tRNAs and other small RNAs. Next question. The transcription is initiated by joining of option 1, 2 free antipedes, option 2, 3 free antipedes, option 3 is 4 free antipedes, and option 4 is 5 free antipedes. And the correct answer is option 1, that is 2 free antipedes, that is nucleotide triphosphates. So the transcription is initiated by joining of 2 free nucleotide triphosphates. The initial binding between the promoter and the polymerase is referred to as closed promoter complex because DNA is not unwound. The polymerase then unwinds 12 to 14 bases of DNA from about -12 to +2 to form an open promoter complex in which single-stranded DNA is available for transcription. This transcription is initiated by joining of 2 free nucleotide triphosphates. Next questions. Which one is incorrect statement regarding polyadenylation? Option 1 is polyadenylation is occurred at the 3' end. Option 2 is at first the pre-mRNA is cleaved at the poly-A site for the polyadenylation signal to begin. Option 3 is after cleavage at the poly-A site, the poly-A-tail is added. Option 4 is poly-A-tail is added by poly-A polymerase, and option 5 is poly-A-tail is between 500 to 600 residues. And the correct answer will be here that poly-A-tail is between 500 to 600 residues. So here the question is you have to choose the incorrect statement. And the incorrect statement is poly-A-tail is between 500 to 600 residues. It is not correct because poly-A-tail is almost 250 nucleotides residue long. That is the option 1, that is polyadenylation occur at the 3' end. It is correct. For polyadenylation to occur, at first the pre-mRNA is cleaved at the poly-A site. It is also correct. After cleavage at poly-A site, poly-A-tail is added. It is also correct. Poly-tail is added by poly-A polymerase. It is also correct. Fifth option is poly-A-tail is between 500 to 600 residues. It is not correct because this poly-A-tail is 250 residues long. Poly-A-tail is almost 250 nucleotide residues long. The polyadenylation protects the pre-mRNA from enzymatic degradation and facilitates nuclear export and translation. So that is polyadenylation protect the pre-mRNA from enzymatic degradation and facilitates nuclear export and translation. Both cleavage and polyadenylation occur at the polyadenylation site, which are located within the 3' untranslated region, that is the 3' UTR. Most eukaryotic genes contains the multiple PAs, that is polyadenylation sites. A conserved hexanucleotide hexameric sequence, that is AAUAAA, occurring upstream of the PS, contains the most important signal for pre-mRNA cleavage and polyadenylation. Which one is correct statement regarding Ruho independent termination? Option is One is termination. In this case, it brought about by the formation of hairpins of newly synthesized RNA. Option 2, a specific protein named Rho bind to the growing RNA chain or weakly to the DNA, and in the bound state it acts as ATPase and terminates transcription. Option 3 is termination codon UAA or UAG bind to the tRNA and stop transcription. Option 4, none of the above. And the answer will be option 1, that is termination in this case is brought about by the formation of hairpin of newly synthesized RNA. So termination of transcription is occurred by two methods. One is Rho dependent termination and another one is Rho independent termination. In Rho dependent termination, a specific protein that is named as Rho factor bind to the growing RNA chain or weakly to the DNA, and in the bound state it acts as ATPase and terminates transcription. So it is the two option. Option 2, that is it is about this Rho dependent termination. So, and in Rho independent termination, the termination in this case is brought about by the formation of hairpins of newly synthesized RNA. When the hairpin loop formation occurs in the RNA transcript, it slows down the polymerase movement, and some pausing is associated with the termination. So, in Rho independent termination is caused by the formation of hairpins of newly synthesized RNA, which slows down the polymerase movement, and such pausing is associated with termination. Next question. Why the 5' cap protects the mRNA from degradation by exonucleases? Option 1 is because the presence of methyl group. Option 2, because of the presence of guanosine residue. Option 3, because of the presence of unusual 5' triphosphate linkage. Option 4, none of the above. And the correct answer is option 3, that is because of the presence of unusual 5' triphosphate linkage. Next question. For nuclear encoded genes, the splicing occurs in the option 1 nucleus, option 2 cytoplasm, option 3 mitochondria, and option 4 is all of the above. And the correct answer is option 1 nucleus. Splicing is a process in molecular biology where a newly made precursor messenger RNA transcript is transformed into mature mRNA. It works by removing all of the introns that in the non-coding region of RNA and splicing back together exons that in the coding regions. For nuclear encoded genes, the splicing occurs in the nucleus either during or immediately after the transcription. Next question. How many types of RNA polymerase in prokaryotes? Option 1 is 1 type, option 2 2 type, option 3 3 types, and option 4 is 4 types. And the correct answer is option 1, that is 1 type. So in case of prokaryote, there are 1 type of RNA polymerase, whereas in case of eukaryotes, there are 3 types of RNA polymerases, that are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. RNA polymerase 1 produce the one transcribed into RNA except the 5S RNA. RNA polymerase 2 transcribe the mRNA, and RNA polymerase 3 transcribe the tRNA, also the 5S RNA, and some small nuclear RNA, small RNAs. Next questions. Which statement regarding the spliceosome is incorrect? Option 1, it is a splicing machinery. Option 2, it helps in the removal of intron. Option 3, it consists of 150 proteins and 5 RNAs. Option 4, it consists of 120 proteins and 6 RNAs. And the correct answer is option 4, that is it consists of 120 proteins and 6 RNAs. Let's discuss about the another options. regarding spliceosome, it is a splicing machinery. It is correct. The spliceosome is a splicing machinery. It helps in removal of intron. It is also correct because spliceosome helps in the removal of intron. Option 3, that is it consists of 150 proteins and 5 RNAs. It is also correct because splicing machinery comprises 150 proteins and 5 RNAs. These 5 RNAs are U1, U2, U4, U5, U6. These are collectively called as small nuclear RNAs. These RNA protein complexes are called small nuclear ribonucleoproteins. The removal of intron is promoted by these small nuclear ribonucleoproteins, that is snRNPs, and called the splicing. Next question. Which statement regarding polycystonic mRNA is incorrect? Option 1. It carries information of several genes. Option 2. It is translated into several protein. Option 3. Most mRNA found in bacteria and archaea are polycystonic. And option 4 is most mRNA found in eukaryotes are polycystonic. So from this 4 options, you have to choose the incorrect option regarding the polycystonic mRNA. And the answer will be option 4, that is most mRNA found in eukaryotes are polycystonic. It is incorrect because most mRNA found in eukaryotes are monocystonic. Whereas about the another options, that is it carries information of several genes, it is correct that polycystonic mRNA carries information of several genes. It is translated into several protein. It is also correct because polycystonic mRNA translated into several proteins. Option 3 is most mRNA found in bacteria and archaea are polycystonic. It is also correct because most mRNA found in bacteria and archaea are polycystonic. Whereas most mRNA found in eukaryotes are monocystonic. An mRNA is said to be monocystonic when it contains genetic information translate only a single protein. Whereas the protein produced by polycystonic mRNA found in bacteria and archaea have related function regulated together in one operon. Next question. Ribozymes are option 1 DNA, option 2 RNA, option 3 protein, and option 4 is none of the above. And the correct answer is option 2, that is RNA. Ribozyme can catalyze their own accession from their parent RNA molecule. Ribozymes, that is ribonucleic acid enzymes, are RNA molecule. They have the ability to catalyze specific biochemical reactions including RNA splicing in gene expression similar to the action of protein enzymes. The most common activities of natural or in vitro evolved ribozymes are cleavage of RNA, DNA and peptide bond formation. Next questions. Presence of which features makes the RNA less stable as compared to DNA? A is presence of free 2' OH group. B is single ring nitrogenous base. C is uracil in place of 5-methyl uracils. So option 1 is A and C, that is free 2' OH and uracil in place of 5'-methyl uracils. Option 2 is A and B, that is free 2' OH and single ring nitrogenous base. Option 3, that is B and C, that is single ring nitrogenous base, uracil in place of methyl uracil. And option 4 is C only, that is uracil in place of 5-methyl uracils. And the correct answer is option 1, that is A and C, that is presence of free 2' OH group and uracil in place of 5-methyl uracils makes the RNA less stable as compared to DNA. Thank you. If you like this video, please like, share and subscribe to this channel to get more videos. Thank you.